Good morning, everyone. In today's session, we will be focusing on the laboratory identification of select metazoa, as we have done for select protozoa, and we will be focusing on the clinical specimen and the stages to be identified for these metazoan parasites. The learning outcomes of this session will start by the identification skills, whereby the students will develop the ability to identify select species of cystodes, trematodes, and nematodes based on morphological egg characteristics that are observed under the microscope. Second, they will be able to learn more about the select diagnostic methods used to detect and identify these metazoan infections in humans. Third, when we cover very briefly the life cycle, students will have a feel of the complexity of certain life cycles of certain metazoa, including their intermediate hosts and the various stages of development. A summary on pathogenesis will be also offered to reinforce what was covered in the lectures. And finally, the students will acquire skills in preparation for the interactive session that we will hold together in pertaining to the identification, diagnosis, collection of specimen, and management of parasitic infection. As such, when we expose specific medical cases dealing with symptoms with clinical specimen, students will be able to identify what was this metazoan parasite. And for later, these developed skills will be crucial for evidence-based decision-making in clinical and public health contexts. Just a quick reminder, if you remember, we divided the parasites according to the number of cells into protozoans, which are the unicellular parasites, and metazoans, which are the multicellular parasites. Metazoans were further divided into helminthes and arthropods. The focus of my lecture today will be about helminthes, which are divided into platyhelminthes or flatworms and nemathelminthes or roundworms. The platyhelminthes can be also divided into cestodes, which are tapeworms or ribbon-like worms, and trematodes, which are flukes or leaf-like worms. For the nematodes, these are the roundworms having a cylindrical and symmetrical body. Let me start talking about cestodes, and just to remind you, cestodes are these flatworms that have basically a ribbon-like shape, and as you can see here, the body is made up of segments that are called proglottids, and the group of these proglottids form a strobila. So I will be selecting some cestodes and talking about their. In this table, you can find a summary of some medically important cestodes regrouping Taenia solium, Taenia saginata, Diphyllobothrium latum, Echinococcus granulosus, Echinococcus multiocularis, Hymenolipis nana, Hymenolipis diminuta, and Dipylidium caninum. For the sake of time, I chose to only talk about four important cestodes, which are Taenia solium and saginata, Diphyllobothrium latum, and cestodes are divided into Cyclophilida and Pseudophilida. Under Cyclophilida, we regroup Taenia, Echinococcus, Hymenolipis, Spirometra, and Dipylidium species. Under Pseudophilida, the single species is Diphyllobothrium latum. If we are to compare now Cyclophilida to Pseudophilida, we have to first start by the scolex, which is the head of the worm. Within the scolex of Cyclophilida, we always find four suckers. For some of them, we find in addition two hooklets, a crown of two hooklets that we call rostellum. In the scolex of Pseudophilida, we find an almond-shaped head that contains two grooves to serve for attachment. If we now look for the proglottids of Cyclophilida versus those of Pseudophilida, the proglottid, which is the segment of the strobila of Cyclophilida, contains uterine lateral branches as well as a lateral genital pore. The proglottid of Pseudophilida contains a central genital organ or a rosette that is central, plus a central genital pore. Of note,
All these cestodes are hermaphroditic, which means that in their proglottids they hold both the male and the female reproductive organs, which means that in the same worm we have the self-fertilization. This is why they are hermaphroditic, and for all cestodes we do not have male worm or female worm. They carry on both genital organs in the same segment. So this is the structure of a cestode or a tapeworm. Again, the head area of the worm is called a scolex that has the four suckers. Here you can see two, but there are two of the other side. Some of them will present the crown of hooklets, which is called the rostellum. You have a small neck that will link the scolex to the different segments that are called proglottids, and the group of proglottids forms the strobila. And towards the end, the proglottids become mature and they will be full of eggs. This is what we call gravid proglottids. So here, this is a microscopy showing the scolex of a cestode. Here you can see the rostellum, and these are the four suckers that allow the attachment of the worm to the specific tissue. The two select cestodes I will be starting with are Tinea saginata and Tinea solium, which I will be discussing together. A quick reminder of the life cycle of these two cestodes. When we ingest infected meat, raw meat or undercooked meat of pork that contains cysticercae of Tinea solium, or cow meat containing cysticercae of Tinea saginata, what happens is that we will have an excistation. We will have basically scoliosis that will attach to the small intestine, and within the small intestine we will have the development of the full worm. Here you can see the scolex, and of course again you will see the strobila of different proglottids. More you move towards the end, more these proglottids are large and gravid because they will become full of eggs. When the cycle continues by shedding within the feces either the egg or the proglottids, the gravid proglottids of either Tinea saginata or Tinea solium, according to the type of ingested meat. Now, when we look at the egg is similar for both Tinea saginata and Tinea solium, whereas for the proglottids they are different, and I'll come back to this later. But to introduce it to you, usually Tinea saginata proglottids are longer with more uterine branches, whereas those of Tinea solium are smaller with less uterine branches. Now, the cycle continues when the cattle or pigs ingest the eggs of Tinea solium or Tinea saginata, cattle for Tinea saginata, pigs for Tinea solium. So upon ingestion of eggs of these parasites in the contaminated vegetation by or even the gravid proglottids also transmit the infection back to the cattle or the pigs. What happens is that we will have the formation of a larval stage that is called an oncosphere. That oncosphere will cross the wall of the intestine of either the cattle or the pig and will go through the blood to the musculature. Within the musculature, these oncospheres will develop into cysticercae, and a new cycle begins when we ingest raw or undercooked meat containing these cysticercae. So how to diagnose Tinea saginata to start with? If you remember on the life cycle, I said that within the feces we can have either the eggs or the gravid proglottids that go out. Now for the beef tapeworm, remember that proglottids are larger and contain more uterine branches than those of Tinea solium. So when we look at the proglottid that is shed in feces of a patient infected with Tinea saginata, we can count around 20 uterine branches on each side. And as you can see, the genital pore is lateral. Now, how can we count these uterine branches? It's very easy. We have to put a drop of ink in the in the genital pore, sorry, and from that genital pore, it will diffuse and label all the branches, and this is how we can count them. So we can count between 15 and 20 branches, and for the sake of simplicity, I will go also for the highest number. So if there is a medical case with these worms, I will be. Now for the pork tapeworm proglottid or Tinea solium proglottid, the proglottid is shorter, is more compact than that of Tinea saginata. We can count between 7 to 13 lateral branches on each side. The genital pore is also lateral. So again, for Tinea solium, the proglottid is shorter, is more compact with. So, as a summary, a slide related to Tinea saginata versus Tinea solium. Let us start with the scoliosis. So, Tinea solium scolex has four suckers and a crown of hooklets, which is the rostellum. It is narrower than that of Tinea saginata. If you look at the scolex of Tinea saginata, it has the four suckers. It lacks the rostellum and is a little bit wider than that of Tinea solium. If we look now at the proglottid of Tinea saginata versus that of Tinea solium, the Tinea saginata proglottid is longer with more uterine branches and a lateral genital pore. That of Tinea solium is shorter, more compact with also a lateral genital. It is worth noting that when we ingest the raw meat containing cysticercae of Tinea saginata or Tinea solium, what we develop is an intestinal disease, and this disease is not dangerous. However, for Tinea solium, we can have another spectrum of disease that happens, and that is called cysticercosis. Cysticercosis differs from the intestinal disease caused by the same worm by the fact that if we ingest contaminated food with eggs or gravid proglottids, what happens in our body is similar to that to that happening in the pork, which is the main transmission species for this parasite. So if accidentally it happens that we ingest food that is contaminated by gravid proglottids or eggs of Tinea solium, what happens is that we will have oncospheres that are hatching in our intestine, boring the wall of the intestine, and then going through the bloodstream to our muscles, our connective tissue, our brain, our lungs, our eyes, and this is where they will develop into cysticercae. So these cysticercae can withstand. Now, whether we are talking about Tinea saginata or Tinea solium eggs, we cannot distinguish or we cannot conclude from the egg which type of Tinea are we seeing. And even for Echinococcus granulosus, we can also see the same feature of eggs. So the egg per se cannot be conclusive for Tinea saginata or solium or Echinococcus granulosus. We would need other indications such as, for example, looking at the proglottids and their length and their uterine branches in case of saginata and solium, and of course for hydatid cysts that I will be covering in a little bit in case of Echinococcus granulosus. Now, for all these tinea eggs, I just want to tell you that the eggs are round, they are they have a shell that is radially striated, as you can see here, and they contain a hexacanth embryo that is inside, which is basically an oncosphere containing 6 hooklets. These hooklets will evolve into basically either the 4 suckers and rostellum, or only the 4 suckers with 2 that degenerate. And these eggs are refractile under microscopy. Let me now tell you about Echinococcus granulosus, which is the tinea of canine or dogs. The difference between Echinococcus granulosus and the other teneas is that the strobila is much shorter than that of Tinea solium or Tinea saginata, and it comprises only three proglottids. Now, if I am to remind you a little bit about the life cycle happens usually between dogs and sheep. Accidentally, we might get the infection. So, the usual life cycle happens whenever a sheep or goat or swine ingests embryonated eggs that are present on the grass they are eating. Now, in the intestine of these sheep, goat, or swine, the oncosphere hatches and penetrates the intestinal wall, and then it will go throughout the circulation to reach other organs, including the liver and the lung, where it will develop into a hydatid cyst. Now, whenever the dog eats contaminated meat from the sheep or the goat or the swine that is contaminated by these hydatid cysts that have scoliosis inside of them, what happens is that the protoscolics from the cyst will protrude, will exist. The scolex will attach then to the intestine of the dog, and it will evolve into an adult that again contains the scolex and only three proglottids. And from there, we will have production of embryonated eggs that are shed in the feces of the infected dog. Now, if it happens that and we ingest embryonated eggs in contaminated food or water, what happens in our body is the same than what happens in sheep or goat, which means that we will have the oncosphere that hatches in our intestine that leaves through the blood and that will transform into hydatid cyst in main vital organs, including the liver, the lung, and also the brain. Of note that these hydatid cysts are full of fluid and they usually contain protoscolices that are attached to the inner layer of the cyst. I will come to the structure and to the features of these cysts in a little bit. However, keep in mind that the liquid that is inside that cyst is very toxic. So if it happens in humans that we need to remove that cyst from a vital organ, we should be very. This is Echinococcus granulosus adult worm. So remember that a main difference between Echinococcus granulosus and Tinea solium or saginata is that the strobila, which is the group of proglottids, is much shorter and consists of only three segments in Echinococcus granulosus, whereas it might be extremely long in case of Tinea solium or saginata. So if we look at Echinococcus granulosus adult worm, we find a scolex that has the four suckers plus the double crown of hooklets making the rostellum. The scolex is followed by a small proglottid that is the immature proglottid, followed by a second mature proglottid, and then comes the third gravid proglottid that is full of eggs, and this is what is liberating the eggs within the feces of infected dogs. So basically, as you can see here, the third proglottid is the gravid proglottid that contains the egg. And this is on your right, the scolex. The scolex has four suckers and it has a rostellum. In that sense, it is similar to the scolex of Tinea solium, but not Tinea saginata. So as a summary, we said that Tinea solium has four suckers and a rostellum. Tinea saginata has only the four suckers. Echinococcus has four suckers and also the rostellum. So these are the differences between the scoliosis of Echinococcus granulosus, Tinea solium, and Tinea saginata. If we go to the proglottids now, Tinea solium and Tinea saginata has much longer strobila with a much higher number of proglottids, whereas Echinococcus granulosus has only three proglottids, an immature, a mature, and a Clinically, what we can see in case of an infection with Echinococcus granulosus is first a hydatid cyst. So what is the hydatid cyst? This cyst is made up of a thick wall that is made up by different layers, including basically a laminated layer that follows a host organ parenchyma, and what is very important is the inner layer, which is called the germinal layer. From this germinal layer, we will have budding of protoscolices. Each one of those will become a scolex and will develop into an adult worm in the intestine of a dog. So if you look back to the hydatid cyst, you will see that this is a hydatid cyst attached to, of course, the host organ parenchyma, whether it is in the liver or in the lung or even in the brain. And inside this hydatid cyst, we find a lot of fluid, and attached to the inner germinal layer are protoscolices. So, for example, in this one, we will have at least 6 worms that will develop from this hydatid cyst, knowing that each protoscolix will evolve into a scolix. And this is another picture of a hydatid cyst. I just want to dwell to your attention the presence of the layers that are very nice here and that are very well seen. This is, for instance, the germinal layer, from which you will have all the protoscolices that will develop. Here you have the hydatid cyst wall, and if you look back, for example, here you can see basically the parenchyma of the organ. And inside, of course, you will have the cyst fluid that can reach several liters in some cases, and this cyst fluid is very toxic and might lead to anaphylactic shock if the cyst is ruptured upon surgical. Now I want to introduce to you a difference between what is a hydatid cyst and a hydatid sand. As long as the protoscolices are attached to the germinal layer, we talk about a hydatid cyst. However, when these protoscolices have broken free from this germinal layer and are floating freely within the fluid of the cyst, the hydatid cyst becomes now a hydatid sand. So here, for example, you can see that by sedimentation, we could see by gravity those protoscolices which are free. They are not attached to any germinal layer, so this is hydatid. Remember that platyhelminthes were divided according to the shape of the worm into either ribbon-like or cestodes, or leaf-like or trematodes. I just finished covering 3 select cestodes. Now I will be moving on to talk about trematodes, which are also called flukes, which are flatworms, and this is the second part of platyhelminthes I will be talking about. Let us remember together the general characteristics of trematodes. So again, these trematodes basically have a flat, fleshy, leaf-like shape. They have 2 suckers, an oral and a ventral sucker, allowing them to attach, the oral sucker being also the beginning of an incomplete digestive system. Remember that for cestodes, there is no beginning, no end of the digestive system. The food gets into the body by phagocytosis or by absorption, whereas the evacuation of waste happens also by fusion to the worm tegument and then exocytosis. Now, if we talk about the trematodes, we will have to start seeing a beginning of the incomplete digestive system. It is incomplete because there is no anus. Also, the waste is evacuated from the worm by exocytosis. Now, if we move on to the reproduction of trematodes, all trematodes, like cestodes, are hermaphroditic, which means that they have both male and female reproductive organ within the same body. There is one exception to that, which is the schistosomes, whereby schistosomes have basically 2 separate sexes, which makes them having male and female worms. And the schistosomes are the transition towards nematodes, which are all having separate sexes. Now, when we talk about trematodes also, I want you to remember that their life cycle is quite complex, and it will require several intermediate hosts in order to develop. If we talk about the general characteristics of their eggs have an operculum, which is a lid that is present at the anterior part of the egg that opens to liberate the larval stage. The only exception to that is also schistosomes, where the egg of schistosomes hatches randomly and is not operculated. So again, for all the trematodes, we have an incomplete digestive system, which starts by an oral sucker. They have all a ventral sucker to attach. They are all hermaphroditic except schistosomes, which have two separate sexes. Their eggs present always an operculum, which is a lid that opens to liberate the larval stage, except also for schistosomas, where we don't see this operculum in the egg. The stages in a typical life cycle of a trematode are quite similar. They all start by having an infective stage, which is the metacercaria. The only exception is schistosoma, and I will come back to this in a little bit. Metacercaria, upon ingestion into humans, they will transform into an adult. The adult will lay unembryonated eggs that, once in contact with water, they become embryonated. There is only one species that might produce embryonated eggs directly. However, generally it is an unembryonated egg which in contact of water becomes embryonated. From the embryonated egg, we will have the liberation of a free living ciliated stage that we call miracidium. This miracidium will swim until finding a mollusk, in which it will evolve into three stages called rhidia, sporocysts, and then cercaria. These cercaria are again flagellated, so they will swim until finding the other appropriate intermediate host, in which they will be transformed into metacercaria, which are infective stages. So this atypical life cycle is similar to all trematodes except for schistosomas, whereby the infective stage for schistosoma is a cercaria. There is no metacercaria in the life cycle of schistosoma. In addition, if you look at the produced egg for all trematodes, it's usually an egg that presents an operculum, which is a lid that is at the anterior part of the egg and that will open to liberate the miracidium stage. For schistosoma, this egg is not operculated and there is a random hatch. So these are again the typical stages in a life cycle of a trematode. So here you can see the operculated egg, except for schistosoma it's non-operculated, from which we will have the ciliated miracidium, that is a free swimming larval stage that swims until finding the appropriate mollusk, in which it will transform into sporocyst, then rhidia, then cercaria. The cercaria is the flagellated stage that is, in case of schistosoma, the infective stage, whereas for the others, this cercaria will transform into another stage, which is metacercaria, in another intermediate host according to the species of trematode. This metacercaria, for most of them, is the infective stage, and upon ingestion of metacercaria there will be formation of the adult, which is the leaf-like structure, which corresponds to the adult of the trematode, that will produce eggs for establishing a. There are different medically important trematodes. These include Phaseolopsis buski, Phaseola hepatica, Clonorchis sinensis, Paragonimus westermani. But the select one I will be talking about, which is exceptional to all other trematodes by several features, is Schistosoma species. So I will be covering in my coming slides as a typical or select fluke the example of schistosomas. Just to remind you, schistosoma is a blood fluke. It requires a snail. It doesn't have metacercaria. Its infective stage is the flagellated cercaria. It lays non-operculated eggs that will hatch randomly to liberate the miracidium. So I will be covering schistosoma as example or as select clinically important trematode. The life cycle of schistosoma starts whenever a cercaria that gets out from snails penetrate the skin of an infected or of a human patient. Whenever it penetrates the skin, it will migrate into the blood and it will lose its tail to become a schistosomole. Within the schistosomole, it will further continue throughout the circulation and migrates to the portal blood in the liver to mature into male and female worms. Remember that all the other trematodes were hermaphroditic. The only exception to that was schistosoma, and the difference is that for schistosoma we see both the male and the female worms. These will produce eggs, and these eggs are different according to species. I will be focusing on that. However, on the life cycle, I will be telling you that eggs of schistosoma mansoni and schistosoma japonicum are usually shed in feces, whereas those of schistosoma hematobium are usually shed in urine. In addition, the egg of schistosoma mansoni presents a sharp lateral spine. That of schistosoma hematobium presents a terminal spine. That of schistosoma japonicum is smaller and more round in shape. Now, upon contact with water, these eggs, which are not operculated, will hatch to release the ciliated free miracidium stage. This miracidium stage will swim until finding the appropriate snail tissue, in which it will evolve into sporocyst, then rhidia, and then from the rhidia we will have the cercaria, which have this V-shaped tail, allowing it to penetrate through the skin, making the cercaria the infective stage. So if we start by just looking at the eggs of schistosoma mansoni, which are the clinically diagnostic stage, eggs of schistosoma mansoni are usually elongated. They are refractile. You cannot miss them because they all have a lateral spine, a sharp lateral spine that distinguishes them from the other schistosoma species. These eggs are shed in the stool and they have a thin shell that surrounds them, and inside, of course, they have the miracidium stage that will be liberated upon contact. If we look now at schistosoma hematobium eggs, these eggs are shed in urine, not in feces. They have a terminal spine that you can see here. This is the terminal spine that is characteristic of schistosoma hematobium. They are also elongated, they are also refractile, and of course they harbor inside the miracidium stage that will be liberated in contact. The eggs of schistosoma japonicum are smaller in size. They are rounder, so they are round, they are smaller, they are also refractile. And unlike schistosoma hematobium or schistosoma mansoni, the spine of schistosoma japonicum is much smaller and is not that clear on all preparations. Here, for instance, you can see it and it's really very small. On this one, for example, you cannot see it. So what is more characteristic of schistosoma japonicum is its round and small shape. Also, it harbors, of course, the miracidium stage that is liberated in contact with water, and the eggs of schistosoma japonicum are like those of schistosoma mansoni, shed in the stool of the infected. As a summary of all schistosoma species eggs and put side by side on the same slide, here is the egg of schistosoma mansoni, which is big in size and which has the sharp lateral spine. Here is the egg of schistosoma hematobium, which is also elongated and big in size with a terminal spine. And here is the egg of schistosoma japonicum, which is more round and smaller in size. Remember that schistosoma mansoni and schistosoma japonicum eggs are shed within the stool, whereas those of schistosoma hematobium are shed within. The last part I will be covering in my talk today is about nematodes, which are round cylindrical worms. What are the general characteristics of nematodes? Nematodes are all round in a cross section. They are bilaterally symmetrical. They have a cylindrical body that is not segmented like that of cestodes. Their sizes can range from 1 millimeter to 1 meter. They have different organs of attachment. Their cuticle is usually thick. They have a very well developed digestive system, which is complete, which starts with a mouth and ends with an anus. And they also have other systems, including a nervous system, an excretory system. They have muscles, and they are sexually dimorphic, which means that all of them have male and female worms. They develop by molting. Usually, they undergo 5 larval stages, and the female is usually larger because it is the one that lays the eggs. This table summarizes some of nematodes of medical importance. These include Enterobius vermicularis, Ascaris lumbricoides, Toxocara canis and cati, Trichuris trichuria, Ankylostoma duodenale, Necator americanus, Strongyloides styrocoralis, Trichinella spiralis, as well as the Filarida, which regroup Wickeraria bancrofti, Brugia malaye, Loa loa, Mansonella species, Onchocerca volvulus, and others. Diagnosis of nematodes depends on whether they are intestinal nematodes or Filarida. Intestinal nematodes usually are characterized by the presence of the adult within the intestine, and their diagnosis is usually made by looking for the eggs that are in the stool specimens. For these eggs, and in order to characterize the intestinal nematode, we should be looking at the color, the thickness of the shell, and we should look for other specialized structures, including plugs, knobs, spines, and opercula. For Filarida, the story is totally different because we don't see eggs. What we have to look for is microfilaria, and these are present either in the blood specimen or in the subcutaneous tissue according to the species of Filarida. The first intestinal nematode I will be talking about is Enterobius vermicularis. And just a quick reminder of the life cycle. A human get infected usually upon ingestion of embryonated eggs. These embryonated eggs will liberate a larval stage within the small intestine. These larval stages will migrate to the colon, where they will become adults. These adults are the male and the female, and as you can see, the female is longer because it is the stage that lays the eggs. So the male and the female will mate within the colon, and the males usually die after mating. The gravid female that is full of eggs will migrate to the anal area, and within the perianal area it will be laying the eggs. After laying the eggs, the female will also die. So when we reach that stage, we will have eggs on the perianal fold that will contain the larvae, and these ones will mature within 4 to 6 hours after being. Please note that Enterobius vermicularis is also called the pinworm, and this is due to the shape of the eggs that you can see here. These eggs are usually round from one side and flattened from the other side. So this is the round side and this is the flattened side, and they are partially embryonated when they are shed. It is worth noting that we do not see these eggs within the stool specimen, and their diagnosis requires of note, people at risk of infection with Enterobius vermicularis are usually kids. We can have outbreaks specifically in daycare or in KGs because this is where children do not pay attention to hygiene and still share food and so on and so forth. So, in order to diagnose these kids, we have to undergo the cellulose scotch tape method, which is applied to the anus of the suspected patient. These samples should be taken the first thing in the morning and before having a shower. The eggs will adhere to the tape, and this is where we can label them and look at them under the microscope. We might need to do more than one swabbing. We might need to do that for 3 consecutive days in order to confirm the infection. The second intestinal nematode I will be talking about is Trichuris trichuria, or also called whipworm, because the adults resemble a whip. So basically, we get infected whenever we ingest embryonated eggs. These will hatch in the small intestine as larval stage. This larval stage will move on to the cecum, and within the cecum, it will evolve into two separate sexes, a female and a male, that will mate and that will produce unembryonated egg that is shed in feces. The unembryonated egg is characteristic and it has a lemon shape or a barrel shape. We cannot miss it in the stool. This unembryonated egg will embryonate in the outside environment until becoming infective while harboring a larval stage inside of it. So here you can see the two adults that resemble a whip. This is why Trichuris trichuria is called a whipworm, and the characteristic of the egg is that it has two poles, and it resembles a lemon. Or the diagnostic stage of Trichuris trichuria or whipworm is again the egg that is shed in feces of infected patients. These eggs are barrel shaped, they are thick shelled, they have a bile staining, and they possess two plugs, so you cannot miss them. These really resemble a lemon, and you cannot miss them under a microscope. These eggs, when passed in the stool, are unembryonated and require a certain period of time in the outside environment to become embryonated and become. The third intestinal nematode I will be talking about is Ascaris lumbricoides, knowing that it has a specific life cycle that requires larval stages to be coughed up and reswallowed before it becomes adult. So basically, just a quick reminder about their life cycle. Ascaris lumbricoides life cycle starts upon ingestion of embryonated eggs. These embryonated eggs in our digestive tract will liberate larval stages in the small intestine. These larval stages will invade the intestinal mucosa and will be carried via the portal, then systemic circulation until reaching the lungs. Within the lungs, they will go from the bronchioles to the bronchi to the trachea, and then from the trachea they will be coughed up, then reswallowed, so they penetrate the pharynx. From the pharynx, they join the digestive tract and go back to the intestine, and this is where they will become male and female worms. Again, the female worm is longer in size as for all the other nematodes because it is the one that lays eggs. Of note, this female can lay two types of eggs, whether unfertilized and fertilized. Only the fertilized eggs will continue in the outside environment to become infective. The unfertilized eggs will end up by being disintegrated. Here you can see the fertilized egg of Ascaris lumbricoid, which is basically the infective type of egg. This is usually a round egg that contains an embryo, and this is one very rare picture showing a larval stage hatching from the egg. The egg presents usually a thick shell with a mammillated layer that is dark stained and usually brown. These fertile egg are smaller than the non-fertilized. And here you can see side by side the picture of a non-fertilized or unfertilized egg on the left and a fertilized egg on the right. As you can see, the unfertilized egg is smaller in size and contains the embryo. The unfertilized egg is oblong, is bigger in size. It is a spongy. It has a spongy feature, and this one does not contain an embryo and gets disintegrated in the outside environment, so it is not infected. Let us now discuss blood and tissue nematodes, and the first example I will be talking about is Trichinella spiralis. So when do we get infected with Trichinella spiralis? It is when we ingest undercooked meat of pork usually, what happens is that the larval stages will be liberated in the small intestine, and they will lead to the formation of adults, male and female, in the small intestine. The larvae will be deposited in mucosa, and then they will leave within the blood circulation to insist again in the striated muscles. So basically, when we are infected with Trichinella spiralis, we will see within our muscles spiral shaped larval stage. This is why we call the worm Trichinella spiralis. It is according to the shape of the larval stages that insist in our muscles. The insisted larval stages of Trichinella spiralis usually are the diagnostic stage. Of course, we need to do a biopsy to see them. They are elongated in shape. You can see them spiral inside the striated muscle as well. And the characteristic of the infection with Trichinella spiralis. The last part of my talk today will be also related to blood and tissue nematodes, but this time I will be covering Filarida, and I will be talking about the life cycle of Wichereria bancrofti, which is usually discussed along with Brugia malariae, knowing that the difference between Wichereria and Brugia is mostly in their geographical locations. So, how do we get infected with Filarida? With, for example, Wichereria bancrofti. This happens whenever the mosquito is taking a blood meal. It will inject into our blood an L3 larval stage. Within the blood, we will have migration of these larval stages until they reach the lymphatics. Within our lymphatics, they will evolve into male and female worms. These will start producing microfilariae. Remember that in Filarida we don't have production of eggs, we rather have production of microfilariae. These microfilariae will be migrating into lymph and blood channels, and whenever the mosquito is taking a new blood meal, it will ingest those microfilariae. These microfilariae will penetrate into the mosquito midgut and then migrate to the thoracic muscles, where they will evolve into an L1, then an L3 larval stage. This L3 larval stage is the infective larval stage that will migrate to the head and then to the mosquito proboscis to undergo or to infect a new patient. The important notions I want you to recall about Filarida are the following. First of all, their diagnostic stage is a microfilaria and not an egg. Second notion is that if you are to diagnose Wichereria bancrofti or Fibrugia malayi, you should take the blood during night time, because these filarida have a nocturnal pattern, and the diagnosis should be done on a thick blood smear taken at night. Now, if I talk about Loa Loa have a diurnal pattern, which means that you need to take the blood from the patient during daytime in order to see these microfilariae. Now, if you look at the three types of microfilariae, I want you to look at the end of these three. Now, all of them are tapered. What happens as difference between Wichereria bancrofti and Brugia malayi is that the end that is tapered for Wichereria bancrofti is usually devoid of nuclei, whereas for Brugia malayi you will find two nuclei, one terminal and one subterminal. If you look at the microfilaria of Loa Loa, the nuclei go till the end, so there is a continuous display of nuclei all over the microfilaria. If I show you now one by one the microfilaria of the three mentioned filarida, let us start by Wichereria bancrofti. So first of all, it is a sheathed microfilaria as for the other two filarida. So it presents a sheath that is outside. You can look here, it has continuous nuclei until reaching the end, and this end is devoid of nuclei, so it doesn't have any nuclei at the end. This is very characteristic of Wichereria bancrofti. Remember that Wichereria bancrofti, to be diagnosed, should be has a nocturnal pattern, so the blood collection should. If we look now at Brugia malayi microfilaria, If you remember, I said that it is also a sheeted microfilaria. This one is probably one of the best pictures I have seen for Brugia malayi. It's very clear. It has continuous nuclei until the end, where we do not see a continuum. However, we can see a subterminal nucleus and a terminal nucleus, and this is what characterizes a microfilaria of Brugia malayi. As for Wichereria bancrofti, Brugia malayi microfilaria have a nocturnal pattern, so in order to be able to diagnose them, we should collect the blood from the patient over. If we look now at the microfilaria of Loa Loa, microfilaria of Loa Loa present continuous pattern of nuclei all over the microfilaria. Microfilaria are also sheeted, but you can see that there is a continuum of nuclei all over the microfilaria, and Loa Loa displays a diurnal pattern. So in order to see these microfilaria, you should collect the blood from the patient during. And I will finish on this slide just as a reminder that not all filarida have sheeted microfilariae. The three first, A, B, and C, are sheeted, so these correspond to Wichereria bancrofti, where the end of the microfilaria is devoid of nuclei. Second, you have Brugia malayi, which is also a sheeted microfilaria with a subterminal and terminal nucleus. Third, we have Loa Loa, which is also a sheeted microfilaria with a continuum of nuclei all over the microfilaria. But if we look at D, E, F, and G, these correspond to Onchocercus volvulus and Mansonella species, and all of these are non-sheeted microfilaria. So I just wanted to remind you that I covered today select members of nematodes, of cystodes, and of trematodes, keeping in mind that these are not the only clinically important ones, but for the sake of time, we had to choose part but not.
So finally, and to wrap up, going back to the learning outcomes, under the first learning outcome, we explored together the eggs of select nematodes, trematodes, and cystodes. For instance, we talked about the characteristics of eggs of schistosoma species, those of Trichuris trichuria, those of Ascaris lumbricoides, as well as Anterobius vermicularis. We also explored the diagnostic techniques, and as such we have included microscopic examination of stool specimen or urine in case of certain schistosoma species, biopsies, for instance Trichinella spiralis, proglottids of tinea, whereby in the segment we put a drop of ink and we count the uterine branches in order to identify the species. And we also covered very briefly life cycles. As such, we talked about the different stages of the life cycle of a trematode. We covered the intermediate hosts and the human host whenever it is pertaining to infections like Enterobius vermicularis or Ascaris lumbricoides, among other parasitic infections. And finally, regarding the fourth learning outcome, we briefly mentioned again each disease and its symptoms for each selected metazoan infection that we covered. And this is for sure reinforcing all the material that was covered in the different parasitology sections by myself or by Dr. Awar. We also, by talking about the life cycle, the symptoms, and the identification of stages in the clinical specimen, prepared the students for the convenient medical decision for therapeutic options later on. With that said, I am done with the lab characterization of select metazoan parasites. We will see others in a face-to-face session where I will be selecting medical cases from the lectures of parasitology, the ones I covered myself or Dr. Awar covered, and we will talk about that in medical cases covering the most important parasitic infections. Thank you so much.